Alright, so I want to talk about active transport, which is different than the other forms of transport that I've talked about so far. The ones I've talked about before are simple diffusion and facilitated diffusion, and osmosis as a specific type of facilitated diffusion. And all of those, as passive transport, happen naturally. They don't require extra energy. Other than the energy that's already in the system, or the kinetic energy of these molecules that are just always bumping into each other, right? So that's the kinetic theory of thermodynamics, right? These molecules are the Brownian motion in chemistry. Are these molecules always moving because they have temperature, which is what we're measuring, right? In temperature, we measure their movement, so they're always going to be bumping into each other. In active transport, we're talking about something very different, and things moving, quote unquote, unnaturally. So this is what is causing that unnatural movement that I'll describe in just a moment. This is ATP. And if you remember from my earlier video, the gray sections are carbons, the blues are nitrogens, the reds are oxygens, and the yellows are the phosphorus. So this is adenosine over here. This is the adenine that we find in a DNA molecule, but when it has this group on it, this is the adenine, the nitrogenous group. This is added to it, which now we call this whole thing an adenosine, and we have our three phosphate groups. So if we had these two covered up, we would have adenosine monophosphate, or AMP. If we had two of them, it's adenosine diphosphate, or ADP. And if we've got all three of them, it's adenosine triphosphate with three phosphate groups, or ATP. So the real cool part of this model is that this part breaks off. And when that bond breaks and we end up with an ADP and a lone P, the energy that held these things together at this covalent bond is released, and we can use it for other processes in our body. And in fact, all living things use that energy that's stored right there. That energy came from the glucose molecule that started the whole process of cellular respiration. And with those hydrogens that got carried through to the electron transport chain, the ATP synthase in the electron transport chain sticks these guys back together. So the ADP and the P wander back to the inner mitochondrial membrane of the mitochondria, and these get snapped back together from that energy, and then this wanders out of the mitochondria to whatever needs it, and then that thing breaks it apart, releasing the energy, and then these guys go back to the mitochondria. Okay? So this ATP is key for this process. So I'm going to pull this out to give you the setup of active transport. So this is a lot messier. There's a lot going on here. Here is my protein. Here's my phospholipid bilayer, and I've got lots of blue dots and purple squares on my screen. The blue dots are sodium ions. The purple squares are potassium ions, K+. This is Na+, K+. They actually have the same charge. They're both positively charged. And so what's kind of neat here to think about is that, or to notice, I didn't really care about my numbers on the left-hand side. The numbers on the inside are really what we're going to follow and track here. So if we counted up all of our things here, we've got 1, 2, 3, 4, 5, 6, 7 sodium ions, and 1, 2, 3, 4, 5, 6, 7, 8 potassium ions. Altogether, that's 15 positive charges. Let's assume we also have 15 positive charges on this side. So then it's at equilibrium. But let's say that the cell is not designed to be at equilibrium. It actually needs a difference or a gradient. And so in order to do that, we need the bad boy of active transport, ATP. So ATP is what comes in and bonds to this protein. And it doesn't work really well here, but this ATP will wander in, it binds to a site on the protein, and it breaks off the last phosphate group. And these guys, this phosphate stays on this protein. This ADP wanders back, heading back to the mitochondria. This P will change the shape of this protein. and we'll actually change the shape to look more like this. And then this phosphate will be released, and the protein jumps back to its original shape. So that phosphate is really important because it's going to actually pump things against a gradient. Now, we are at an equilibrium, so it's not actually against a gradient. It's going to create a gradient. So we see that this is open, and when it's open, these three slots are going to be filled. These three sodium spots. So I'm going to go ahead and fill these in. So this is going to come in, this is going to come in, this is going to come in. Now these look like they're open, but they're not really open. That's the limitation of this model, that we can't just go ahead and jam stuff in there. But our ATP, so 3 sodium go in, our ATP comes in, that it gets phosphorylated, this phosphate group gets added, the protein changes shape, the sodium come out, so we get 3 additional sodium on the outside. So remember, we start with 15 positive charges, now we're down to 12. And let's say there were 15 out here, now we're down up to 18. Well, these spots are now open for potassium ions. The phosphate group comes off, wanders back to the mitochondria to bring back, to combine again by the ATP synthase at the electron transport chain into more ATP. But when that phosphate comes out, it changes the shape of this protein again, kicking out these 2 positive charge ions to the inside of the cell. Now if we were to count the positive charges on this side, we'd end up with less. Let's look at this. Remember, the blue dots are the sodium and the potassium. Also, they both have positive charges. So let's count them together. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. I'm down 1, and I'll end up with 1 extra on the outside. That's because 3 sodium went out, 3 positively charged sodium went out, and only 2 positively charged potassiums came in. So it's 3 out, 2 in. That's going to cause a gradient. Let's do it again and see what happens. So I'm going to have 3 more sodiums coming in. Oh, you couldn't see all of them before. All right, 3 more sodiums coming in. We've got another ATP molecule coming in, phosphorylating the shape of that protein, changing the shape of it, kicking out these 3 sodiums, opening up for 2 potassiums, releasing the phosphate group, changing the shape of the protein back, releasing these to the inside. And now I've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 positive charges on the inside. So again, I'm down. So there are less positives on the inside than on the outside. And that's key because now I have a gradient. Even though we're only talking about positive charges, there are fewer of them here than here. So we can say that this is relatively negative to this relative positive. Even though we're only talking about positive ions here, this is relatively negative and this side is relatively positive. Okay? And active transport continues in this way, but as you can imagine, there's going to be a natural gradient. And sometimes what happens is we get a protein that's somewhere else on this phospholipid bilayer that changes its shape when there's enough of a charge that's different. And when there's enough of a charge, it opens up, letting some of these sodiums flood back in. And which is kind of crazy, is thinking, what's the point of all of this? This is the key to, one example of this is that this is the key to your nervous system and how your nerves send signals. And we're going to learn a little bit more detail about that in the near future, specifically about your nerves. Okay? And that's active transport. And it's obviously going to keep going against the gradient. And in order to do that, from going from low concentration to high concentration, it needs the ATP, the energy from ATP. Okay, it needs energy from this molecule. All right? I think that's sufficient. I hope you have questions and that you add those to your need to know next steps document so that I can answer them for you and for anybody else who has questions about this.